Hello and welcome back to Simply Biology. In today's video we're going to be looking at the formation of tissue fluid. We're going to be looking at why it is important, how it can be formed and what role it serves in the body. So without further ado, let's get started. So tissue fluid is formed by the high hydrostatic pressure at the arterial end of the capillary. So remember, you've got the two ends. You've got the arterial end, which is next to the artery, which connects the artery, the capillary. Then we have the venule, which connects the capillary to the vein. And what happens is that due to the high compressions and the pressure changes of the heart, causing the smooth muscle and elastic fibers to expand and recoil in the arteries, this causes a high hydrostatic pressure at the arterial end of the capillary, and this forces water and other dissolved nutrients out of the blood plasma. Remember, the blood plasma is the liquid part of the blood, approximately consists of 45% of the blood. And this is forced out, and this is what's known as tissue fluid. And tissue fluid is useful because it helps to bathe all the cells in the body. This therefore allows the exchange of materials with every cell, all cells in the body. So how tissue fluid is able to pass through the extremely thin one cell thin layer known as squamous epithelial cells and the small holes in the capillary wall known as fenestrations. The large plasma proteins, however, and blood cells do not leave the blood along with the other nutrients. These create a high oncotic pressure at the venule end of the capillary. Therefore, oncotic pressure is now greater than hydrostatic pressure. As a result, this creates a net inward force, and this draws all the fluid back into the capillary. So remember, you've got the high hydrostatic pressure at the arterial end of the capillary. This creates a net outward force, forces dissolved nutrients and water out. This allows exchange with all the cells in the body. However, large plasma proteins and blood cells are too large to fit through the squamous epithelial cells and the fenestrations in the capillary wall. So therefore, these do not leave the bloodstream. They create what's known as a higher oncotic pressure. Oncotic pressure becomes greater than hydrostatic pressure at the venule end of the capillaries, and this creates a net inward force and draws the fluid back into the capillary. However, not all of the tissue fluid is drawn back into the capillary, but the excess drains into what's known as the lymphatic system, into blunt-end lymph nodes. Once the tissue fluid enters the lymphatic system, it becomes known as lymph, and it's then returned to the bloodstream via the blood vessels. What happens is you've got the artery, falls into the arteriole, which then goes into the network of capillaries. Then you have the venule, which connects the capillaries to the veins. So there's a higher hydrostatic pressure here at the arterial end of the capillary and a lower hydrostatic pressure here. The high hydrostatic pressure forces the dissolved nutrients and water out and allows the exchange with all these cells in the body. And then at the venule end, there's a lower hydrostatic pressure and a higher oncotic pressure. So this helps to make sure all the dissolved nutrients or all the waste products that have been exchanged with the cells are absorbed and pulled back into the bloodstream, where they go off into the veins and return to be disposed of. Here we also see what's known as the lymph vessel with a blunt-ended lymph node, and remember, when the tissue fluid drains back into the lymphatic system, it becomes known as lymph. Well, thank you for watching this video, and I hope you found it really useful and beneficial, and it helped you explain the concept of what tissue fluid is, and I hope you've managed to understand the uses of it in the body. Thank you, and goodbye.